Okay, welcome back. We are going to be talking about the process of protein synthesis in this video. The combination of physiological processes that accomplish protein synthesis are transcription and translation. So we're going to take a look at transcription and translation today in this video. I want to begin by just saying that your cells and the cells of all living things, you can think of them as protein generating factories. That's basically what the function of every cell is. It is to create proteins that are helping the organism complete the functions of life. Just as an example, when you're extracting energy out of a meal, it requires dozens and dozens of digestive enzymes. All of those enzymes are proteins, and those proteins have to be made by your cells. So the cells in your pancreas, for example, and cells in other parts of the body, for maybe the stomach lining, are producing these enzymes on a regular basis, and that process is called protein synthesis. So that's just one example. Your muscle cells also function only because of the proteins that are in them, actin and myosin, that interact with one another. Those proteins have to be synthesized. And so the cell is essentially that factory that produces those important proteins that help you do those functions of life. In a very general way, if we step through the processes that are involved in transcription and translation, the first phase is just understanding where in the cell it's happening. So first you want to find out, well, where is the blueprint with all the information necessary to learn how or know how to produce a particular protein. And so, you've already studied in a previous chapter what a protein is, and it's a sequence of amino acids linked together into a long chain that folds back on itself and eventually makes a three-dimensional structure. But where's the instructions for how to build that? And so those instructions are found in your DNA. So within the nucleus, in every cell, is housed the information for how to build every protein in our bodies, including those digestive enzymes I talked about, including those muscle fibers that I talked about, and just on and on. All of the proteins in your body, the information is within your DNA. That information on a very specific basis is known as a gene. So when you talk about a gene, this is the best definition, or the simplest definition of a gene, is just that it's the blueprint for how to build a protein, or at least components of a protein. And so the process of protein synthesis begins inside of the nucleus. And it's specifically in the DNA. So if we click on that, you'll see the DNA. Remember, those are the chromosomes. There's where your genes are, inside of the nucleus. And that's where the process begins. We're going to do transcription inside of the nucleus. The product of transcription, you will find, then is shipped out of the nucleus and into the cytoplasm. Once it's in the cytoplasm, that product, which we call a messenger RNA, is going to join up with a ribosome. And it's on these ribosomes, either attached to the endoplasmic reticulum or free floating in the cytoplasm, where the process of translation begins. And the translation is to take the information in the messenger RNA, which has been coded right off of the gene. You'll see this in a minute. And to turn that into a sequence of amino acids, which becomes your protein. So it's these two processes, transcription first, which happens inside the nucleus, and then translation, which happens out in the cytoplasm, that collectively create your protein for you. So the process of transcription, and we said this occurs inside of the nucleus, is depicted here in this cartoon. And what you can see, first of all, is the gene, the DNA strand, or the chromosome. The gene is one little tiny bit of that long chromosome, is depicted here in this DNA strand. And you can see that what's happening is that the DNA strand is broken apart, kind of unzipped, very much like a zipper. Just unzip it. And then, using the nucleotide sequence here on the chromosome, right? Using that nucleotide sequence, you build a mirror image of it in a new molecule. Rather than DNA, you're building a new molecule in what's called RNA. Notice over here, messenger RNA, or a transcript, is created. And so notice here, the DNA transcript is like this. It's saying, here's my DNA, thymine, guanine, cytosine, adenine, again, thymine. And what's happening is you're going to be putting down the exact partner for that, the the partner that joins together. And as you create these base pairs, you get a mirror image of the gene, but that's the same information. So wherever T is, you get A. Wherever G is, you get C. Wherever C is, you get G. Wherever A is. In this case, you don't get T because RNA has uracil rather than thymine, but still, this information is an exact coding of what's on your DNA. So it's kind of like copying the blueprint of how to create the protein onto this product, and then eventually taking this product, which we call the messenger RNA, all right, and it separates from the DNA, and then you ship it out of the nucleus through those nuclear pores and into the cytoplasm. So this is the process that's known as protein synthesis. This is known as transcription. So let's watch this video which briefly describes the process of transcription.
All cellular protein building and much of the cellular gene expression occurs through the two principal steps called transcription and translation. In transcription, DNA's information is encoded or transcribed into RNA. For genes that code for proteins, the RNA is called messenger, or mRNA. The process begins when RNA polymerase opens the double helix of DNA, like the slider on a zipper. It unwinds the DNA double helix and pries the two DNA strands apart. As the RNA polymerase continues down the length of the DNA, several actions take place. It reads one strand of the DNA and transcribes the DNA's information. It creates an mRNA version of the DNA, or transcript. The mRNA transcript is a complementary copy of the DNA sequence. When the transcription ends and the mRNA transcript is complete, it separates from the DNA template and exits the nucleus through a nuclear pore. The transcript enters the cytoplasm on its way to the next step in the process of polypeptide synthesis, translation.
Okay, so now our messenger RNA, or also known as the transcript, has been photocopied. Essentially, think about this. It's been copied right off of the gene. It's bringing that information out into the cytoplasm and saying, hey, here are the instructions about how to assemble your protein. And remember, your protein is just a unique sequence of amino acids linked together, and that will fold on itself and create a specific protein. And there are 20 different amino acids in the body, in every living organism, as a matter of fact. And so you can imagine the unique sequence you can do if you're adding tens of thousands of these amino acids along in a sequence, which is a typical number in a big protein. The variety is just endless for how you can arrange these. And so that's why we get so many different kinds of proteins in the body. But the instructions, again, for that is going to be in the transcript. So here's the messenger RNA transcript that we made in the nucleus that is going to be translated into a protein. And the way this is done, and I'm going to keep this very simple, is that it is read 3 nucleotides at a time. So these 3 nucleotides, they could be, for example, A, U, C. And these 3, that could be G, G, C, or C, U, C, or whatever they might be. They're literally read 3 at a time, where each of those 3 nucleotides corresponds to a single amino acid. And I'm not going to ask you to go through and learn all those. The 3 together are called a codon, C-O-D-O-N. And so the codon is what is actually being read and saying, okay, these 3 together, this codon codes for a particular amino acid. Now, if you look at this, the larger structure here, along with the smaller structure here, together is a ribosome. And so the ribosome has 2 subunits. It has a larger subunit, which you can see here above, and a smaller subunit down here. And they kind of sandwich the transcript in between the 2. And the transcript kind of moves through it like a ticker tape. And as it does, it exposes these 3 nucleotides, the codon, to a spot where a new kind of RNA, this is called transfer RNA right here, this gray thing, can come in, and notice it has the anticodon right here on it. So what it does is it finds the codon, matches up perfectly, and that particular transfer RNA can only carry one specific amino acid, whatever corresponds to that codon. So on the transcript, if it says, here's GGC, that only codes for one amino acid. This transfer RNA that has that particular amino acid has the perfect anticodon to fit it. Again, if this was GGC, this would be CCG. It has to base pair, just exactly the same way all nucleotides base pair. So that's the way the cell knows exactly which amino acid to bring next. And then imagine the ticker tape moves forward. These three come into place here, bringing a new transfer RNA with a new amino acid. And this creates this perfect string of amino acids with exactly the instructions that were in your genes. So I do have a little video to watch of that, maybe a little more complex than we need, but let's take a look at it. Okay, as we prepare to look at that video, let's just remember, here we have a transcript being made in the nucleus, shipped out of the nuclear pore into the cytoplasm, messenger RNA out in the cytoplasm. Here it is, having joined together with a small subunit of the ribosome. A larger subunit will eventually settle down on this. But the thing to notice here is the codon here, as I said, those three nucleotides that give you the unique information to tell exactly which amino acid we want to put in this place, and the anticodon that does it. So this tRNA has a specific anticodon. Let's say again, this might be GCU. This would be CGA. Again, the exact, the anticodon will fit right onto the codon, bring a very specific amino acid into place. Okay? Now let's watch this video.
Translation begins when the mRNA enters the cytoplasm and a small ribosomal subunit attaches itself to the end of the mRNA strand. At the same time, a transfer RNA, tRNA, molecule recognizes and binds to the start codon on the mRNA strand. tRNA recognizes the mRNA codon through its anticodon, which has a sequence complementary to the mRNA codon. It is the tRNA molecules that carry the amino acid building blocks of the protein. Next, a large ribosomal subunit joins and forms the fully functional ribosome that will join the amino acids on the tRNAs to synthesize the protein. When the amino acids of the two tRNA molecules are joined, the tRNA in the first position is released to get a new amino acid. The growing protein is attached to the tRNA in the second position. The mRNA is then pulled through the ribosome so that the tRNA...
I just wanted to notice how each time a new codon, three nucleotides, pulls into the ribosome, a new tRNA discovers that. The anticodon fits perfectly, bringing a unique amino acid. So let's start this again.
With the protein chain is now in the first position. Now, a new tRNA with the proper anticodon can come into the ribosome at the open position. This pattern continues as tRNA after tRNA bind to the mRNA codons and their amino acids are added to the growing protein chain. Toward the end of the mRNA sequence, the ribosome gets to a stop codon, which does not code for any amino acid. This signals the end of the protein and that the translation process is complete. The ribosome then releases the finished protein from the tRNA and detaches from the mRNA as the two ribosomal subunits separate. The protein will then be further modified in other cellular structures or directly used by the body.
Generally, those other structures they're talking about there, which modify the protein, is a combination of the endoplasmic reticulum. Many of these proteins are going to be made inside of the lumen of the endoplasmic reticulum. Remember those ribosomes that are studded on that? That's what they do. They create the proteins, and the proteins are actually on the inside of the ER. At that point, it will be housed into a little vesicle that gets transported out to the Golgi apparatus. And when it fuses with the Golgi apparatus, those proteins, which are kind of proto-proteins, not quite finished yet, get finished inside of the Golgi apparatus. And then you've got a finished protein that can either be shipped out of the cell or used inside the cell. So that's pretty much transcription and translation in a nutshell.